So let's look at now the humoral immunity. So of course, the humoral immunity is run by B cells, the B lymphocytes, and the B lymphocytes we saw. First of all, admit someone. Okay. So the B lymphocytes, as they are, we saw that they perform two roles. They first of all are antigen presenting cells, but they also produce antibodies. They produce antibodies. And antibodies are what we refer to as immunoglobulins. Refer to them as immunoglobulins. And therefore, antibodies are abbreviated with IG, meaning immunoglobulin. Okay. So we have different types of antibodies, and they are abbreviated IG. IG to mean immunoglobulin. Yes, we know that there are types of proteins, and these proteins are globuline. They are globular, they are a bit soluble in water. Yeah. And so, of course, the term humoral immunity comes from a term, humus, that means body fluids, right? That is what it means. So that is where this terminology is coming from. But for us to know that just the humoral immunity is run by B cells, and it's run by B cells because B cells are producing antibodies, or what we refer to as immunoglobulins. So what usually happens is that when an antibody, when a B cell encounters the antigen, when a B cell encounters an antigen, of course, the antigen is presented to a T cell. That is one thing that usually happens. Then once it's presented to a T cell, the T cell is going to interact with the B cell via the T cell receptor. If you remember what we talked about yesterday, we said that T cell receptors and B cell receptors are ideally in the same family. It's called the immunoglobulin supergene family, meaning they are similar. They look like immunoglobulins. And one thing we noted yesterday is that immunoglobulins are Y-shaped. They are Y-shaped, and they are of different types. So we look at some of the immunoglobulins, not so much in details, but we'll be able to understand the different types of immunoglobulins. So a T cell has encountered an antigen, okay? So it will interact with a B cell via the T cell receptor. Once that interaction happens, now the B cell becomes terminally differentiated, and it ceases to be called a B cell. It now becomes a plasma cell. And so this plasma cell is able to synthesize antibodies with the help of Th2 helper lymphocytes. And just like Precious has told us, Th2 are going to secrete interleukin 4 as well as interleukin 10 for then these particular antibodies to be synthesized. So antigen presenting, of course, we know that antigens are either processed via MHC2 if they are exogenous, or MHC1 if they are endogenous. So, but in this case, we are talking about Th2 helping Th2 cells helping antibodies, helping B cells in antibody production, which means the antigen presentation is being done now in MHC2 complexes. And once that happens, then CD4s, which are the Th2, are going to secrete interleukin 4. They're also going to secrete interleukin 10, and that then will bring about immunoglobulin production. Okay, so initially, what happens is that there is the production of IgM. There is the production of IgM. So IgM is immunoglobulin mu. It is the largest immunoglobulin. I think yesterday Amas talked about it. We said it has a valency of 10. So let me just see if I can get yesterday's slides and flash that. Okay, so I believe you're all seeing this. So IgM is what is initially produced. So you see, IgM is like 5 antibodies stuck together via the J chain. They are stuck together via the J chain, like that. So you see, of course, this is the FAB region. The yellow parts are the FAB regions, the regions that interact with antibodies. The regions, the regions that interact with antigens, sorry, the fragments that interact with antigens are this in yellow. So you see, this is a very large molecule. It's called immunoglobulin mu. This is the first antibody that is generated. It is the first antibody that is generated. Then later on, there is something we call class switching. There is class switching depending on the invading pathogen, depending on the location of the invader. There will be class switching. Now, you see, this is a broad immunoglobulin. It is large like this. And the reason for it being large, it's a bit non-specific, so it's able to bind as many antigens as possible because it has a very large valency, as you see. So it's able to trap as many antibodies as possible. Before then, specializations come into place. Yes. Okay, I think you are able to now see why IgM is the first antibody that is generated because it has a large valency like that. Okay, then so there will be class switching to either IgG, IgA, or IgE based on the type of antigen and the location. Based on the type of antigen and the location. Now, if the antigen or the pathogen, in this case, is found either in blood or tissues, then you are going to switch to IgG. If it's found in mucosal surfaces, then it's going to be IgA. If it's found on surfaces, it's going to be IgE. If it's again found in either blood or lymph, it's going to be IgM. IgM will be maintained. So it depends. Depends with the site and the type of antibody antigen that is infecting the body. So there is class switching. And of course, we have memory B cells. These ones are usually maintained. The plasma cells are maintained and they're able to survive. Some can be able to survive forever. Some can take a shorter time. And the essence for even booster vaccinations is so that then you're able to build a secondary immune response as quickly as possible. We know that we have infections that you can only encounter and suffer once in life. And any subsequent encounter with the antigens are quickly dealt with because the memory B cells are there and they've been kept forever. We have some that are able to survive only for a few years, and so you require boosters. You require boosters for you to be able to then eliminate. And that's the essence for vaccination. So as nurses, I'm sure you'll be dealing with lots and lots of EPI. It's called the expanded EPI, yeah. Kenya Expanded Program on Immunization, the EPI, KP. So you'll be monitoring kids, of course, from childbirth all the way. We also have maternal immunizations that are supposed to take care of the unborn child. So those vaccinations, of course, as you get into the clinical years, you'll be able to follow through and see when are boosters given, why is it supposed to be done at that particular point in time. All right. So maybe I just forgot to include IgE here. I forgot to include IgE, and I've said that IgE will mainly deal with respiratory antigens and antigens that are found maybe in a respiratory surface, and situations of hypersensitivity also. IgE will be class switching will happen so that then there is IgE production. All right. And again, we need to remind ourselves that we have 5 types of immunoglobulins. We have 5 types of immunoglobulins. So we have IgM, or IgM stands for immunoglobulin mu, IgG, which stands for immunoglobulin gamma, IgA, which stands for immunoglobulin alpha, IgE, which stands for immunoglobulin epsilon, and lastly, we have IgD, which stands for immunoglobulin delta. But you don't see it anywhere here. But in a moment, we'll see what it does. So it might not be directly being produced by antibodies to neutralize any antigen, but it plays a key role. It really plays a key role in the process of antibody production. We'll see that. Okay. So what do B lymphocytes do? What do they do? We know what they do. We know that what they do, and B lymphocytes are either going to be antigen presenting cells or they are going to produce immunoglobulins. And they have the ability to produce 5 types of immunoglobulins. Uh, of these 5 types of immunoglobulins, that is IgM, IgG, IgA, IgE, and IgD. IgG, if you remember yesterday, the diagram I've just shown you a few minutes ago, IgA has other subtypes. IgG has other subtypes. So we have IgG1, IgG2, IgG3, and IgG4. So you see where it is supposed to be dealing with antigens that are in the blood or in the tissues. And as the antigen evolves, as the infection evolves, especially in the cases of malaria, IgG is usually the one that is produced, and it's able to evolve all the way from IgG1 to IgG4. But in most cases, IgG3 is the one that ultimately eliminates the parasites in the case of malaria. So it depends with the infectious agent, it depends with the location where it's found, and so class switching happens based on that, right? So of course, B lymphocytes are able to produce immunoglobulins, but they're also antigen presenting cells as we saw yesterday. Now, when you look at the B cell receptor, because we said we have a B cell also has its own receptor. So immunoglobulins are on the surface. We have immunoglobulins on the surface of the B cell so that they're able to recognize antigens. Okay. So apart from having antibodies being generated just in the blood systemically in form of IgM, in form of IgG, in form of IgA, in form of IgE, these are being generated systemically so that they neutralize the antigens. We are saying that on the surface of B cells, we have immunoglobulins, monomeric IgM and IgD. Okay, monomeric IgM and IgD. Could anybody tell me what we mean by monomeric IgM? Yeah, Miguel?
I think monomeric IgM is a single unit from the IgM found on B cell surface, acting as
Sorry?
a B cell receptor. Like
Sorry? Yes?
I'm
I
saying
think,
monomeric IgM
Mm.
is a single unit from the IgM found on the B cell surface, acting as a B cell receptor for antigen recognition.
Okay, that is right. Because M generally is found as pentameric. As you see, these are 5 units of uh, the antibody. These are 5 units joined together by the J chain. They are 5. So this is a pentameric unit. So one of these is what we are referring to as a monomeric IgM, and it's found on the surface of the cells. Similarly, IgD is found on the surface of B cells. So on B cells, you're going to encounter IgM, but not in this form, monomeric 1. Then you're going to encounter IgD, And we've seen that IgD is not being generated for any other purpose or to neutralize any antigen. It is just being generated for the purposes of being on the surface of B cells, and so that it's able to attach to antigens and to ensure that antibodies are being generated. So that's the point that I want us to have clearly. IgD, yes, it's produced, but it doesn't play a role in as far as neutralizing um, antigens is concerned. The role it plays is for the recognitions of antigen, so that then antibodies are produced in that sense. So that is what it means here. So B cell receptors, together with IgM and IgD, the monomeric IgM as well as IgD, are able to then now interact and ensure that antibodies are produced. So of course, anti-processing and presentation to CD4 T cells enhances B cell activation. The moment an antigen is presented, first of all, it's processed. So it's processed, meaning it's digested and its peptides are carried on the groove of the major histocompatibility complex. Then, of course, it's then presented to the T cells, in this case, CD4 T cells, and we are talking about Th2 here in this case. Once that happens, then there will be proliferation and differentiation of B cells. Remember, these B cells were naive. They are referred to as naive before they encounter the antigen. Once they encounter the antigen, they are now ceased from being naive. They move to germinal centers, that is the spleen and lymph nodes. They now mature from there and start secreting antibodies. They are therefore referred to as plasma cells. They are no longer being referred to as B cells. In this case, they become plasma cells. So they continue generating antibodies against a specific antigen. So these antibodies are either against several epitopes or specific epitopes of the antigen. Okay, so this secretion of specific immunoglobulins for the recognized antigen, and of course, there's also memory. Memory B cells or memory plasma cells are maintained in these germinal centers, either in the spleen or in the lymph nodes. They are maintained, and therefore, any subsequent infection or vaccination would boost the rest, so that then there is elimination. Of course, and B cells have to interact for this to happen. And B cells interaction is in three ways. The interaction is in three ways. First of all, we know that B cells can act as antigen presenting cells, that we know. So since B cells act as antigen presenting cells, they're able to present antigens on MHC2 to CD4 T cells, to helper T cells. And these helper T cells are specifically the Th2 helper T cells. Okay. Then the interaction between T and B cells can lead to cytokine modulation, in which T cells are able to generate cytokines like interferon gamma. Remember yesterday I said that interferon gamma is a pro-inflammatory cytokine. It's a major pro-inflammatory cytokine. So interferon gamma will then trigger cell differentiation or cell proliferation and differentiation. So for this then to be enhanced, of course, in addition to interferon gamma, interleukin 4 and interleukin 10 also play a key role in assisting the cell proliferation, differentiation, and eventually the production. But these cytokines, first of all, so the T cells start by producing interferon gamma initially to ensure that cell proliferation and differentiation occurs. Then later on, these T cells start generating interleukin 4 and interleukin 10. And this will play a role in as much as they assist in cell proliferation and differentiation, will also play a role in class switching. So there is sequential class switching to ensure production of either IgM. So of course, initially we've seen that IgM is the one that is produced. Once IgM is produced, then there will be class switching to either IgG or IgA or IgE. And this is facilitated by the interaction between T and B cells. So T and B cells, of course, are interacting via the T cell receptor and via the B cell receptor. That is where the interaction is happening. As this interaction is happening, so you remember, if it's antigen presentation, B cells are interacting with T cells, but they require costimulatory molecules. They require costimulatory molecules. Can somebody remind us the costimulatory molecules that we talked about yesterday in terms of antigen presentation? There are two. There are two markers that are important in antigen presentation. There are two, CD what and CD what? Valerie?
CD28 and CD80.
Yes. So those are so that is how they interact. If it's antigen presentation, then of course in this case, other than the B and T cell receptors interacting, there is co-stimulatory molecules that are involved, CD28 and CD80. Then of course cytokine production, seeing that T cells, especially Th1 cells, are going to generate interferon gamma. Th2 cells are going to generate IL4 and IL10. There is then class switching depending on where the infection is. Okay, so that is the result of interaction between T and B cells.